of a speaker who had all of us mesmerized, I think, last time round with his wonderful talk, which was earlier in the series of these talks on sleep, regarding the unsung aspects of sleep. So today, I welcome Dr. Joy Desai, sir, who is the Director and HOD, Department of Neurology, Jaslok Hospital, Mumbai, for his next talk in this series on sleep. The topic for today's this webinar, early in the morning, the fine morning webinar, is Glymphatic Dysfunction, Glymphatic with a G, Sleep, Aging, and Alzheimer's Risk. Over to you, Joy, sir.
Right. Thank you so much, and I'm glad to be back. Let me start my screen share. Can you see the screen now?
Not
yet. Not yet.
Yeah, let me just check. Share screen. There you go. Share.
Yes.
All right.
See now.
Can you see that now?
Yes.
Yeah.
Super. And I'm going to make the video box a little small at my end so that I can see my slides clearly. And I'm happy to start. Okay, good morning, everyone. It's good to be back, and I hope you enjoy this little journey in some discoveries in neurobiology as much as I've enjoyed learning these things. So in June 2013, a researcher who was not well known till then, except in a few circles, Mike Nedegard, took the world of science by storm when she published this paper in Science, and she very provocatively titled it The Garbage Truck of the Brain. And the world of sleep neuroscience ceased to be the same after this publication. And very soon, within about 4 months from the initial paper, from the NIH, they published another little snippet which implied that the brain may have an excretory system that functions exclusively in sleep, and one of the key purposes of this excretory system may be to flush out toxins, which at that time they did not specify the toxins, and later many other experiments in the next 6 years showed that this included phosphorylated tau and beta amyloid. So the traditional concept that we all learned when we were in medical school and a neurology training is that the cerebrospinal fluid which bathes the brain has a typical pathway of its genesis within the choroid plexus. You can see that in the lower box here. You can see the epithelioid cells and the capillary within, and the fenestrations in the endothelial wall of the capillary. And there is a complex physiological process which results in the formulation of CSF, which first has access to the ventricles of the brain, and through the CSF pathways, it has access to the cerebrospinal fluid. Now, what is shown in the box at the top, that this traditional concept has undergone a change. You can see that box within the parenchyma of the brain, and what it shows there is the pericyte of the astrocyte, that is the foot process of the astrocyte, which is encircling the penetrating capillary. And you can see in black what is written there as shown as the diagrammatic representation of aquaporin 4. And the new concept is that the interstitium of the brain expands in sleep, and this expansion is due to a change in the architecture of what is shown here in green as the astrocyte. The astrocyte seems to contract, and when we are sleeping, the interstitium expands. And the purpose of this expansion of the interstitium is to initiate a CSF flow which is unidirectional across the interstitium. And that led to the concept of the glymphatics. And in a nutshell, if one wants to understand that, these researchers implied that there is a unidirectional flow of CSF along the interstitium, which is mediated by this glymphatic system. It occurs exclusively in sleep, and it abates on arousal. And this abeyance or the cessation of glymphatics is mediated by adrenergic tone, that goes up when we waken. They also showed in further experiments that the supine posture and the right lateral posture optimizes glymphatic function, and the prone posture reduces efficiency of this glymphatics by about 40%. Further experiments in mice demonstrated that voluntary exercise and low dose alcohol can enhance glymphatic efficiency, and head injury, diabetes mellitus, lacunar strokes especially, and obstructive sleep apnea could impair the efficiency of glymphatic function. And it was also demonstrated in numerous studies that glymphatics seems to excrete, amongst other things, beta amyloid and tau in normal asymptomatic individuals. So we'll have a look at some of these finer nuances and then study the literature that exists in support of these mechanisms that we have discussed. So this diagram shows you in a very simplistic but illuminative manner. What you see here on the left is a box that shows the penetrating capillary, arterial capillary. What's shown on the right side of the box in blue is the capillary, is the venule. And in between is the interstitium of the brain. And what is shown here is that with the onset of sleep and the expansion of the interstitium, the astrocyte is responsible for the polarization of aquaporin 4, and we'll come to polarization in a few minutes. And it's this aquaporin 4 that imparts the directionality of CSF flow from venule into, from the arteriole into the venule. And that's demonstrated in a 3D representation in the box on the right. So it's not very difficult to understand. So the expansion of this interstitium occurs by almost 40 to 60% during sleep due to changes in the retractability of the astrocyte. The presence of aquaporin 4 on the foot process is a dynamic and modulable polarization. That means the aquaporin 4 can move from the foot process to the soma or close to the soma of the astrocyte during various physiological and aberrant dynamics in the brain. So it is modulable by aging, it is modulable by exercise, and it's modulable by alcohol, and we'll come to that in a minute. What is very clear is that aquaporin 4 is responsible for the directionality of CSF, and that right lateral and the supine posture optimizes this process of glymphatics. And this seems to be related to the fact that in the right lateral posture, this has been shown in mice, the sympathetic tone is the lowest, and we know that an increase in sympathetic tone can seize the lymphatic efficiency and can alter it. And on awakening, the same thing happens, lymphatic ceases, and this seems to be driven by an increase in epinephrine as soon as we wake up. The same group, Mike and Nedegard, published this paper in mice where they showed that perhaps low dose alcohol, and in mice, the dosing of alcohol was taken as 15 ml per kilogram for low dose and 30 ml per kilogram for high dose. And they didn't make the mouse drink the alcohol. They actually injected the alcohol into the abdomen of these mice. So perhaps this data from mice can be taken with a pinch of salt, but it was very interesting what was discovered. The researchers seem to imply that widespread astrogliosis, and if you have a subcellular localization of aquaporin, then it perturbs the lymphatic clearance. And in the CNS, aquaporin 4 is exclusively expressed in the foot process with a very high polarization in the foot process. So if the brain is traumatized, for example, then the polarization of this aquaporin can shift from the foot process closer to the soma, and that can alter the lymphatic efficiency. The same thing seems to happen with moderate doses of alcohol. It increases GFAP expression, glial fibrillary acid protein expression. It lowers the polarity of aquaporin 4 from the astrocyte closer to the soma, and therefore moderate doses of alcohol significantly suppresses lymphatic function. And the reason these changes occur is the influence of alcohol itself on these microstructural changes in the brain. So as far as I could glean from the large amount of data that is available, though they showed that low-dose alcohol can be better, as shown in the middle part of this diagram, where the glymphatic efficiency is shown with a much larger arrow as shown in the right. But if you can see with medium amounts of alcohol, this process decreases and lymphatic clearance falls. So perhaps alcohol is not really recommended as a strategy to improve lymphatic function. And there is a large body of literature which seems to imply that though slow wave sleep initially increases in the first half of sleep after alcohol consumption, it is been shown in numerous studies that in the latter half of the night after 1 o'clock, if one has consumed alcohol prior to sleep, then in the latter half there is frequent fragmentation of sleep and frequent awakening, and the depth of sleep tends to suffer. So slow wave sleep tends to suffer, and there is some increase in REM sleep. It's also very clear that circadian disruption is a feature of alcohol binges. And this seems to be worse in people who are homeless, who have other factors that determine this. And if one wants to summarize the amount of data that is available, it seems very clear that alcohol perhaps is more detrimental to sleep physiology than a proponent of sleep efficiency. Okay, so why is this linked to perhaps the risk of Alzheimer's disease? Now we know that we all learned about neuropathology of Alzheimer. We know that there is an extracellular deposition of senile plaques. This consists of clumped beta amyloid core surrounded by neuritic debris, reactive astrocytes, and what is called activated microglia. And we'll discuss activated microglia in further slides to come. It is associated with an intracellular accumulation of helical filaments of hyperphosphorylated tau. This twists into a coil, and this is known as the traditional neurofibrillary tangle described by Alois Alzheimer. And Alzheimer also described that there is loss of synapse, there is neuronal atrophy, and this produces loss of function, and this is paralleled by an amyloidogenic angiopathy, which is called, he called it congophilic angiopathy, because of the affinity of amyloid in the vessel for Congo red, that stain used to demonstrate amyloid in the arteries. And it's very clear that lobar topography of these changes determine the sequence of presenting symptoms with which a patient can present to a neurology clinic. Now, what is intriguing is that the deposition of tau and amyloid seems to have slightly different topographic evolution in a brain that indolently progresses from mild cognitive dysfunction to severe one. And that's shown here in this diagram. And you can see here that the deposition of tau on the left of the diagram, demonstrated in green, begins in the locus coeruleus and the entorhinal cortex, and then it proceeds from the mesial frontal anatomical areas into the dorsolateral frontal lobes and the parietal cortex as an inside-out phenomenon. On the opposite side, if you look at beta amyloid, it's demonstrated here in the color purple on the right side of the diagram. Beta amyloid seems to get deposited initially in the parietal cortex and in the neocortices of the brain, and then it progresses from an outward inward into the mesial frontal cortex, into the deeper brain structures. And right now, it's not very clear how one can explain the relevance of this disparate pathophysiological process. Perhaps further research will show how this is linked to other nuances of sleep dysfunction. So what is this beta amyloid hypothesis? In a nutshell, we know that the amyloid precursor protein is first cleaved by a beta secretase into a large soluble ectodomain and a C-terminal peptide. And this is further cleaved by a gamma secretase into A beta oligomers, mainly A beta 40 and 42. We have numerous lines of evidence which show that the A beta oligomers 4042 are neurotoxic and responsible for neural degeneration, synapse disruption, dendritic disruption, and cognitive dysfunction. And this process in focal areas of neuraxis like the nucleus basalis of Maynard results in depletion of acetylcholine as a neurotransmitter. And we have some therapies that are aimed at improving the levels of acetylcholine as a part of symptomatic therapy in our patients with Alzheimer's disease. And this diagram shows you a expression in the form of a linear cartoon where the amyloid precursor protein is initially cleaved by the beta secretase, and then gamma secretase produces the A beta oligomers. So this amyloid beta hypothesis was the holy grail of research for a long time until it got challenged by other nuances. With the advent of data from familial Alzheimer's disease and the biology of the four genes, APP gene, presenilin 1, presenilin 2, the ApoE epsilon 4 polymorphisms, and that changed the focus of research. And subsequently, for a period of time, a lot of research focused on phosphorylation of tau and its regulation by this key enzyme, glycogen synthase kinase beta, and the influence of insulin and diabetes mellitus. So for about 10 years or so, from 2008 onwards, when this paper was published in neurology, in a large cohort of about 6,500 odd participants who took part in it. Essentially, this study showed that if you have central obesity and you have an enlarged sagittal abdominal diameter and your BMI is high, if you follow up these people for 20 years, then those with a larger abdominal obesity and high BMI seem to be at a higher risk of developing cognitive dysfunction of the Alzheimer type. And this led to a large body of research driven in England at Denmark Hill by Simon Lovestone, where there was a hypothesis that diabetes itself produces insulin resistance within the brain as well as in peripheral tissue. And this downregulates the activity of GSK3 beta 3 as shown here in the diagram, which has a regulatory effect on phosphorylation of tau. This mounted a lot of studies to study insulin as a therapeutic molecule by nasal insufflation in patients with Alzheimer's disease. And what is shown here is that if you have an insulin resistant state, it upregulates GSK3 beta. GSK3 beta, which is an inhibitor of phosphorylation of tau, can no longer prevent the accumulation of neurofibrillary tangles. And it is this accumulating tau that produces neuronal dysfunction. At the same time, there were other processes of study which demonstrated that diabetes itself is a chronic inflammatory state, not only in peripheral organs, but also within the brain. And this hyperglycemia and its associated HPA axis dysregulation alters the process of oxidative stress, changes hippocampal neuroplasticity, and it's responsible for impaired hippocampal functioning, as shown here, producing depression and cognitive impairment. So in a summary, there is a large body of data which demonstrates that diabetes can impair hippocampal neurogenesis and it can alter the efficiency of hippocampal functioning. And we will discuss that in the light of sleep in time to come. Also, Jeffrey Ellip's study with Michael Nedegard has shown that as the human brain ages, the paravascular clearance pathway of the glymphatics also changes. And what is shown in this paper, which I would recommend for people who are interested, they should read this. What they have demonstrated is that with aging, there is an astrogliosis in the brain that increases. It is associated with a loss of paravascular aquaporin 4 expression, and the localization and the polarization of aquaporin 4 in the foot process changes in an aging brain and tends to migrate closer to the soma. And this polarization change of aquaporin 4 is significantly associated with an impairment of the glymphatics in an aging human brain. So the next question that they asked is, why does this happen? And one of the risk factors for this is also diabetes. It's been shown that diabetes also produces the same change. So if you are aging and you have diabetes, this process only seems to get worse. So this is the diagrammatic representation of what they call diabetes-induced dementia and its close links to altered aquaporin 4 functioning and a secondary impairment in glymphatic efficiency in diabetes. Now, we learned in my first lecture that one of the key functions of sleep is to reactivate waking memories and to consolidate them by a complex process of electrical changes that occur in slow wave sleep. We discussed in the first lecture the nesting of slow wave oscillations with spindles and hippocampal ripples in slow wave sleep, producing consolidation of memory, and we discussed that this process requires a bidirectional prefrontal hippocampal dynamics driven mainly in sleep. Now, this process can also change as we age because in an older population, there is a longer sleep latency. There is a greater number of transition to lighter stages of sleep and wakefulness. There is more time spent awake after sleep onset, and there is a greater fragmented sleep. And we also know that in older brains, the slow wave sleep, especially N3 sleep, which is vital for this memory consolidation process, is impaired and a lesser time is spent in N3 sleep in the early phases of sleep. And I showed this diagram in the first lecture as the change in time across a lifespan with the time spent awakening, the time spent in REM sleep, and the change in slow wave sleep as humans grow older. So in summary, we know that in an older brain, there is an uncoupling that occurs in sleep because of change in slow wave and spindle coupling and prefrontal cortex, especially medial prefrontal cortex atrophy in older individuals. And this has been shown by Bryce Mander and his colleagues and Matthew Walker. It's shown here that as you grow older, the bar on the left in orange is of younger adults, the bar on the right in blue is of older adults, and that shows the scattergram of prefrontal atrophy in the aging brain. And this prefrontal atrophy, as shown here, it's very interesting. There are numerous studies done by this group from Berkeley, and what is shown here in the lower diagram in orange on the left is that prefrontal cortex and the middle frontal gyrus atrophy in older people is associated with slow wave sleep change, as shown here in the density of slow waves being impaired. And if you look at the hippocampal cortex and if you look at the corpus callosum, is shown in the diagram on the right, the change in density and volume of these structures in an older brain impairs the efficiency of sleep spindling, and that's shown in the bar chart here with the green showing that in young and the orange in the older adults, and you can see the drop in spindle density, and on the left you can see the drop in slow wave activity. So it appears that aging decreases the expansibility of interstitial CSF compartment with a reduced retraction of astrocytes, which compromises ICF compartment expansion in sleep. The polarization of aquaporin 4 from the foot process of the astrocyte changes with a localization closer to the soma. It's called the subsoma polarization. And diabetes, binging with alcohol, head injury, especially if recurrent, can accelerate this process. Poor pulsatility of arteries, sleep disordered breathing, and erratic respiratory excursions worsens glymphatic efficiency due to an impaired flow dynamics of the CSF. Remember that in the initial studies in the glymphatics, it was shown that arterial pulsatility and the gentle pulsations of rhythmic respiration in sleep also augment the directionality of glymphatic function. So cardiovascular exercise, maybe low dose alcohol, and I'm not sure about that. Endurance training seems to have the opposite effects on astrocytes, and it improves glymphatic function. There is some evidence that meditation perhaps could also do that, but the jury is not fully out. There is a very interesting study that was done by the Chinese and published in Frontiers in Molecular Neuroscience in 2017 in mice models where you put a mouse in a Morris water maze and make the mouse do exercise for about 6 kilometers equivalent before it gets to the cheese. So in these mice models, in voluntary wheel running in the water maze, as it was shown, the investigators from China demonstrated that exercise produced a number of activated astrocytes, and the activated microglia decreased with exercise. Aquaporin 4 expression increased, and the distribution of astrocytic aquaporin 4 was rearranged with a greater polarization in the food process. In mice, this was associated with an improvement in glymphatic clearance and a decrease in A beta accumulation, and dendrites, dendritic spines, and postsynaptic density also improved. So in mice, it is very clear that voluntary exercise improves glymphatic function in almost every aspect of the molecular physiology that drives this complex process. What is the evidence in humans? We have some indirect evidence. Indirect evidence, as shown in this paper published in Neurology 2017, if you took people who slept poorly and looked at their PSG data and their sleep quality and daytime somnolence, and you measured their CSF for the markers of Alzheimer's disease, CSF A beta 42 and CSF tau, in their self-reported sleep analysis, in those whose sleep was poorer, they had a greater amount of CSF tau and a lower quantity of CSF A beta 42, very typically what happens in Alzheimer's disease in cognitively normal adults. In another study, there was this very cogent paper which was aptly titled "A Restless Night Makes for a Rising Tide of Amyloid in the Brain." If you sleep-deprived individuals both in an acute model and in a chronic model, and you suppress due to sleep deprivation slow waves in sleep, then in such people, the CSF levels of tau and A beta mimic what is seen in Alzheimer's disease, which tells us that in normal individuals on a day-to-day basis, sleep is responsible for clearing amyloid A beta oligomers and tau in normal people. There was a lot of interest in the study of activated microglia, and it is believed that microglial activation and aging are related, and a lot of studies showed that microglia can mediate synapse loss in Alzheimer's disease. And just a few months ago, about 6 or 8 months ago, there was a seminal paper published by Andrew Lim correlating the effects of sleep fragmentation and microglial aging and the risk of Alzheimer's disease. Prior to that, the same group showed that sleep fragmentation in older adults seems to correlate with a lower cortical gray matter volume in the mesial and inferior frontal gyri of these individuals. This was published in Sleep 2016. And subsequently, they published a seminal paper in Neuroscience where they took a large cohort of individuals from the Rush Memory and Aging Project and the Religious Orders Study, and they could show with actigraphy that in individuals who demonstrated sleep fragmentation, there was a higher expression of genes which characterize activated microglia and morphologically aged microglia, independent of their chronological age. And in these people, in those who developed cognitive impairment, and in some of them who died, when their brains were studied for Alzheimer pathology, they found a correlation with worse cognition and with the typical pathology of Alzheimer's disease. So this study, which was also emphasized in Neurology Today as news of the month in last October, there is a clear demonstration that sleep fragmentation alters the physiology of microglia, and microglia, which have the physiological function of cerebral protection from oxidative injury, get activated and start harming the brain and contribute to the pathology of dendritic and synapse loss in these brains. And this relates to the decreasing sleep efficiency as we age. And we also know that this process in someone who develops Alzheimer's disease can get worse and it can have a cascading effect. One of the interesting studies demonstrated that arterial pulsatility, which contributes to glymphatic efficiency, gets altered with amyloid angiopathy because of change in the stiffness of this congophilic angiopathy that accompanies Alzheimer, and it further worsens the glymphatic efficiency. So what I would like to put to you is that there may be a case for considering sleep hygiene assessment in patients with dementia. While the current therapies swivel around symptomatic management, and often the emphasis is on neuropsychiatric manifestation, regulation by medications, and memory dysfunctions seems to be deeply entrenched in these people. The elephant in the room is perhaps sleep hygiene, and this needs better evaluation. And there may be multiple mechanistic contributors to the altered physiology of sleep in a brain rendered vulnerable by age and burdened by poor sleep hygiene and the systemic effects of diabetes in those who unfortunately have diabetes. So prefrontal areas of the brain atrophy, slow wave oscillations and spindles in sleep suffer, sleep efficiency and depth of sleep worsens with age. This makes physiological memory acquisition and retention poorer in simply elder people. The lack of exercise, the presence of diabetes, atherosclerotic stiffening of vessels, sleep disordered breathing, and sleep fragmentation makes glymphatic functioning worse and increases amyloid burden. Numerous mechanistic anomalies in glymphatic functioning, for example, astrogliosis and aquaporin 4 polarization, makes clearance of amyloid and tau poor in the aging brain. And hence, altered sleep physiology perhaps is an inadvertent catalyst in a cognitively failing brain. And how is it so? There is a very interesting study that was done fairly recently, a couple of years ago. Unfortunately, this was published from Australia in a not very prominent journal called Translational Psychiatry. But the point of the study was very interesting. This group, led by Stephanie Rainey Smith, looked at genetic variation in aquaporin 4 and the relationship to sleep and beta amyloid burden. So they studied cognitively normal older individuals from an Australian cohort. They looked at aquaporin 4 genetic variations. They looked at their sleep parameters from their sleep quality index. And in many of these individuals, they performed a PET scan with the Pittsburgh compound for labeling beta amyloid in the brain. So 462 normal individuals participated. They underwent amyloid imaging with the Pittsburgh compound, and they looked at single nucleotide polymorphism variations in aquaporin 4. And what was found was very interesting. They found 3 aquaporin 4 SNPs that interact with sleep duration and had a moderating effect on amyloid beta. 2 of these, their numbers are shown here, were associated with poor sleep quality and a moderation in the relationship between sleep latency and amyloid burden, and carriage of a minor allele was associated with a higher A beta burden and a shorter duration of sleep. What was startling in this study is that in some patients with a peculiar minor allele in aquaporin 4, even longer sleep duration was associated with a higher burden. And this needs further study. And I'm sure that in the next 2-3 years, we will hear more about this. So it's very clear that aquaporin 4 perhaps is emerging as the linchpin of sleep health in people. We know that in voluntary exercise in humans, there is an association of improvement in hippocampal volume and mesial frontal cortex volume, a greater functional connectivity, and this has been demonstrated in various studies. There is also a study correlating these effects in Tai Chi in elderly adults. And very simply put, it seems that your brain loves the gym or the sidewalk or the bike trail or the pool, and therefore exercise seems to have a moderating effect. There are also some initial studies, and shown here I've brought you three different studies which demonstrate the effect of mindfulness and meditation on neuroplastic change in the prefrontal cortex network in elderly people, and is shown here in these diagrams on the left with functional PET imaging. One of the studies is from the United States, the second study is from Korea, and this seems to be an emerging field, and I'm sure people like Dr. Shraddha Shah will find interest in the morphological improvements that occur in the hippocampi and the prefrontal cortices with meditation. So the prefrontal cortex, the health of which has important regulatory influences on memory and emotional salience, as we learned in lecture 1, is susceptible to trauma, aging, alcohol binges, and depression, as shown on the left, where the prefrontal cortex can atrophy, but it is amenable to modulation and neuroplasticity by exercise, mindfulness, yoga, enthusiasm, and passion, wherein perhaps the volumes can increase, as shown on the right. So let me show you a case. This is a 76-year-old homemaker. She's the mother of a prominent anesthesiologist in South Mumbai. She presented with a 3-year history of progressive impairment in immediate memory, episodic confusion, one episode of getting lost while out shopping, and a recent change in behavior with the dominant apathy. She had been treated for hypothyroidism and diabetes. She did not believe in exercise. There was no family history of dementia. On the bedside testing, she exhibited signs of temporal, parietal, and mild frontal lobe dysfunction. There were no extrapyramidal signs when I saw her. And when we got her PET scan done at Jaslok, this is what the PET showed. Typical changes compatible with perfusion anomalies that we see in Alzheimer's disease. What you can see here is bilateral parietal hypoperfusion, bilateral neocortical and mesotemporal hypoperfusion with a typical pattern that one sees as shown here on a different scale, but demonstrating the same findings, very typical of Alzheimer's disease, and it was so reported by the nuclear medicine department at Jaslok. Her husband affirmed that she had been a snorer since the time he knew her, about 40 years. He also said that she could doze off in the daytime at the least pretext, was often fatigued, which he attributed to sedentariness. When I got her polysomnogram done, her sleep efficiency was 86%. She exhibited severe features of sleep apnea. Her REM AHI was 70, her non-REM AHI was 56, and she had a total respiratory disturbance index of 57. On 10 centimeters of water during her titration, her apnea-hypopnea index resolved to less than 3, and she was prescribed rivastigmine, memantine, and CPAP. And that brings us to the emerging notion that perhaps Alzheimer's disease and sleep apnea can in some ways have a shared pathophysiology, and it is believed that in sleep apnea, as shown in the features on the right, intermittent hypoxia, decreased glymphatic clearance, and a alteration in microglial activation due to frequent fragmentation of sleep, as shown by Andrew Lim and his colleagues, and a disruption of slow wave activity would be responsible for the alterations in A beta and hyperphosphorylated tau, which characterizes what happens in Alzheimer's disease. Is this notion somewhat new? The answer is no. I show you this study in Journal of Clinical Sleep, published in Sleep Medicine 2009, where in a small cohort of people, sustained use of sleep app decreased the deterioration of cognition, sleep, and mood in patients with Alzheimer's disease. In a fairly recent study published just about 2 years ago, in cognitively normal elderly people who had sleep apnea before they had developed Alzheimer's disease, when sleep parameters were correlated with Pittsburgh compound imaging of the brain, there was a clear correlation between a higher occurrence of amyloid burden on PIB imaging and the presence of OSA. So there may be a room to treat such people at an early stage, even when they are cognitively normal, perhaps to prevent their downward slide into cognitive impairment. So is that the whole story of Alzheimer's disease? Well, not really. Very recently, there was this interest in periodontal health and the occurrence of Alzheimer's disease. And this took the research world by storm. It was detected that patients with Alzheimer's disease in the brain, they have in the hippocampi and the mesial frontal cortex DNA of this organism called Porphinomonas gingivalis, an organism that's responsible for gingivitis in elderly people. So what the researchers did was they took the bacterium, and in mice, they injected the bacterium into the brain, and lo and behold, these mice brain then went on to develop amyloidogenic changes similar to Alzheimer's disease. So that led to a plethora of lab experiments, and the Japanese came up with this hypothesis that Porphyromonas gingivalis in the host is responsible for the production of 5 or 6 different biochemical products. They've called them gingipains, and 2 of these gingipains in the lab has shown to be a modulator of GSK3 beta, just like diabetes, and perhaps it is in some way responsible for a contribution to Alzheimer in some vulnerable people. So at last we have something which seems to be not related to sleep. Is that really so? Well, this study seems to imply that sleep also seems to have a bearing on poor oral health. And I'm pretty certain that in the next 2-3 years, we will see a lot of focus on the research in the molecular mechanisms wherein poor sleep and oral health can be related. And perhaps in future, we will know how these different aspects get tied up. So there are some clear unanswered questions. In a brain rendered vulnerable by genes like presenilin 1 or ApoE epsilon polymorphism dual haplotype, lifestyle, poor education, trauma, and diabetes, we do not know where in the timeline of life does sleep become the catalyst for a biological cascade of deterioration in the life story of Alzheimer. Many critics argue that Alzheimer itself disrupts sleep neurotransmitter biology, and that sleep dysfunction in Alzheimer is an epiphenomenon. But we have a large body of literature that seems to imply that the sleep dysfunction begins much earlier. We have numerous lines of evidence which suggest that neuroplasticity persists throughout a lifetime, but with declining efficiency. Emerging data does position lifestyle modulation as a preventive intervention. And with that, I'm going to stop here, and this photograph is for Rahul Kulkarni, if he's listening in to this talk. Thank you very much for your attention. I'm happy to take questions at the end of this. Thank you.
Hello.
Hello?
Yeah.
Yeah.
Sir, that was an excellent lecture. So it was not just physical awakening, but also some kind of an enlightening experience for all of us.
Thank you so much.
So awakening in true sense. If you allow, I can take some questions.
Yes, yes, of course. Do you want me to put on the video, or how does this work?
Sir, can I please share my screen so that the questions?
Yeah, let's do that. Do that. So,
you
shall I click stop share for me?
You have to stop sharing, sir.
Yeah, I've done stop sharing. That's good. Okay, cool. We are online. Yeah, Ashish, you're ready to go.
Is this screen?
Right. Okay, so these are the questions.
Yes, sir.
Yeah, can you show that to me again? Yeah.
Is it visible?
Yeah. So first question is, why is it called glymphatics? Well,
Yes,
it comes from 2 words. Glia, because traditionally what we learned when we were in medical school 25 years ago is that astrocytes and microglia are called glial cells, and the term glia was attributed to these cells because it was believed at that time that their main function was to stick the neurons together and work as a scaffolding in the brain. Now we know that the brain does not have a lymphatic system, and therefore when this excretory system was discovered, it was labeled glial lymphatic, and they merged the two words together. It was called glymphatic. Well, subsequent to this discovery of the glymphatics, there is another group of researchers who have also discovered that there is a meningeal lymphatic system that also exists in the brain. And we also know in some other studies, which was the point of controversy about the function of the glymphatics, is that in some experiments in mice, the unidirectional CSF flow, a part of it seems to reflux back in the brain. So it appears that the interstitium is like a cauldron of splashing around CSF, which to a large extent moves from the arterioles to the venules, but a part of it seems to slosh back, only to be sloshed back into the venules if we sleep further. Okay,
So the
so
next one is the
the next one
perivascular
is
space
perivascular space, the same as the
as
glymphatic
glymphatic.
system. So perivascular space in the diagram that I showed you is bound by the so-called glia limitans, is a contributor to the anatomical framework within which the glymphatic system functions. And if you look at the diagram carefully, there is in white what is shown the perivascular space. And there is some emerging information to perhaps show that in some patients who have obstructive sleep apnea, there is a morphological alteration in this perivascular space. And we don't know the true implications of that finding, but I have read some papers which clearly seems to demonstrate that. Okay, the third
Yes, sir, I
question
think
is aquaporin 4 and NMO correlation with glymphatics and the likelihood of cognitive decline. To be really honest, I don't have a very good answer to that question. But I do know, if you remember, if you've listened to all my other colleagues who often talk about NMO, I think Shripad is there in the audience. There is one phenotype of NMO, which is the diencephalic phenotype of NMO, wherein some patients with neuromyelitis optica actually present with narcolepsy-like disorder, wherein their imaging shows the presence of morphological changes within the hypothalamus and the diencephalon in the region that is responsible for the genesis of orexin. And that's where we know that there is some overlap. I am not sure whether in the long run NMO can result in cognitive dysfunction, but I think that may be something that is worth studying.
I think there is similar question on that. If you see the last line in this slide,
Yeah.
if NMO causes astrocyte injury to dorsal medulla, is that the cause of sleep dysfunction? And if it is causing astrocytopathy, why not all NMO patients have sleep problems? I think you said diencephalic variant.
Yes, so I told you that there is a
Could you please
diencephalic phenotype of NMO, we know that. Do patients who have NMO, if they don't have sleep-related dysfunction, do they demonstrate abnormalities on PSG? I think somebody should take this up. And if it is not being done, I'm not sure in NMO literature, because often neuroimmunologists don't seem to talk to sleep specialists. But if someone is interested, this would be a study worthwhile doing. My DNB student, Aditya, I'm sure Aditya is
Hello. I think screen is frozen. Hello.
Yeah, I can hear you, Ashish. I think it's something at Sir's end.
Sir's and I cannot, it looks like frozen. Hello, Joy sir. Joy sir. Joy, sir? I think the connection is
So
lost.
that's a very relevant question. And this has been studied by some researchers. There is a very good publication that appeared about 4 or 5 months ago in one of the sleep journals on the altered glymphatic dynamics in NPH. And there is one study where the glymphatic function in humans has been evaluated with a new kind of tracer, which is different from gadolinium. I think they call it gladobutron. And they have demonstrated that there is an alteration in glymphatic function in NPH. How this differs from the other aberrant patterns in the glymphatic function in patients with OSA and sleep fragmentation, that is something that more research will tell us.
Sir, I'm sorry, your connection had some glitches. So I'm afraid the earlier question was missed by all of us.
Yeah.
The second last question on the screen.
Yeah.
I think just now you spoke about NPH, but the previous question about
Is there more
and
sleep equal to
hypersomnolence,
more waste clearance?
I think we missed that.
Yeah. Okay. So see, the concept of hypersomnolence is subjective to hypersomnolence when. So when we say hypersomnolence during sleep in patients who perhaps are sleeping greater than 8 hours at night, in the traditional hypersomnolent individuals, there are two categories. We have someone who are hypersomnolent due to what looks like a Klein-Devine syndrome, which is a specific entity of dysfunction within the basal temporal cortices of individuals who have hyperphagia, hypersomnolence, and increased sexuality. It's a very specific syndrome. But if one looks at increased sleep in the daytime, usually excessive daytime drowsiness is a manifestation of dysfunctional sleep at night. So we know that the scoring systems for excessive daytime drowsiness are often correlated with aberrant sleep physiology at night, and they are correlated with the presence or absence of sleep dysfunction, which could be either sleep fragmentation, poor sleep efficiency, with or without sleep apnea. So I'm not so sure that a greater sleep beyond the physiologically accepted norm is associated with greater clearance. I don't have a great deal of understanding of that. So I'm not in a position to answer that question.
So the next one, the non-breathing related causes of sleep disruption, or let's say sleep fragmentation,
Yeah.
and the risk of dementia.
Yeah. So can you hear me now? Yeah.
Yes, sir.
So
Yes, sir.
the paper by Andrew Lin is the first major publication showing a very distinct correlation of these two aspects. I'm pretty certain that this was published about 6 months ago, and this came out. I think I shared the data with Dr. Arun Shah sometime in October when we discussed this, and back and forth. And this is something that is likely to become a subject of future research. All of these other questions that you're talking about, arterial pulsatility, circadian rhythm disorders, and antihypertensives, I think it stands to derivation that somewhere a correlate will exist between uh, aberrances in these physiologies and its relationship to sleep health. Some of it we know, a lot of it is unknown. And if we look carefully, I'm reasonably certain we will find somewhere correlations. And the reason why, as neurologists, we should be interested in this is because if we don't take interest in these physiologies, more work cannot be done in understanding the complex relationship between cognition and sleep health.
Right. So the further question about natural sleep versus drug-based or drug-induced, hypnotic-induced sleep.
Yeah.
What is the effect on glymphatics?
I don't have any knowledge on that subject, so I'm not able to answer that question.
And all those patients who are having cognitive decline, do you advise sleep detailed evaluation by history and then polysomnography to look for any reversible causes?
Well,
for
I
cognitive.
don't know. I have started this as an exercise. As I told you, Aditya Onkar, who's a student, is doing this. I've been doing this for the last 2 years now. And perhaps if Dr. Anuradha is also listening in here, she will remember many of the patients that I have done with her. Uh, Clearly, there is a correlation. So we are collecting all this data in patients. And if we have more than about 200 patients now. So somewhere this is going to become the norm, and I think it's worthwhile looking at this.
Okay, sir. So the next one, all those patients diabetic middle-aged, the glycemic control, good amount of exercises, and good amount of sleep. Would it prevent the dementia risk?
Well, we
Should
don't know,
that be
but
the
there is
mantra of
some,
treatment?
yeah, so there is some data to demonstrate that this could be the case. Uh, if you really see patients who are diabetic have, I tried to show you the experimental evidence and the evidence in humans that diabetic individuals, it's been demonstrated since many years, have a systemic inflammatory state that makes a systemic inflammatory cellular vulnerability in almost every organ system. We now have some evidence to show that such is the case with the brain as well. Whether these interventions will decrease their risk, um, as a hypothesis, probably yes, but it needs to be demonstrated. I'm not sure of good studies which have proven this, but that's the hypothesis. So again, if studies are done, I'm sure we will be able to find that out.
Sir, you showed one case where the PET scan was done. Do you routinely advise PET scan for Alzheimer's disease?
Yes, so that is FDG PET. Now, if you really see the evaluation of patients with cognitive impairment, whom you are trying to categorize into, say, minimal cognitive impairment or based on neuropsychological evaluation, overt Alzheimer's disease, and so on, or frontotemporal dementia, then there are various modalities of evaluation. The first is clinical, then there is the biochemistry and systemic risk factors which are contributors like hypothyroidism, homocysteine, etc. Then there can be neuropsychological evaluation for deriving a cognitive picture of the type of cognitive impairment. In the Western world, in research studies, people do CSF with biomarkers. There are three currently accepted biomarkers: the levels of beta amyloid oligomers A4042, the presence of phosphorylated tau, which have an inverse relationship with cognition in the CSF, and the presence of a molecule called neurogranin. Unfortunately, all these three biomarkers are not available in clinical practice today. The third test is an FDG PET of the brain. Now, only in research, people are doing two kinds of pathological imaging, that is PET imaging for tau and PET imaging for beta amyloid with the Pittsburgh compound. In India right now, these two imaging technologies are not available in commercial practice, but we do have FDG PET. So if you do FDG PET, then there are certain very typical patterns which help to differentiate to some extent, not absolutely, but to some extent between frontotemporal dementia, diffuse Lewy body disease, for example, and Alzheimer's disease. And if you ask people like Dr. Vikram Lele and other nuclear medicine specialists, in well-defined clinical phenotypes, the FDG PET shows well-defined patterns. But in some with overlaps, sometimes there is a little blurring of what the FDG PET shows.
Right. Sir, you told about P. gingivalis and gingipains, so and poor oral health. So if you stretch it further, what about the entire gut or gut association? Nowadays, there is a lot of gut-brain
Yes,
links.
so that's an interesting question. Um, there is some ongoing work to look at the gut microbiome and its relationship to sleep health. I am not aware of any strong correlative data that has emerged from this. There is also interest which my other colleagues, Dr. Wadia and Pankaj and many of the others, movement disorder specialists who are doing a lot of work in this, will know that there is an association between gut pathophysiology and microbiome and the initiation of synuclein in the gut. Uh, we don't have very good data to make any strong take-home derivations. And we'll have to study this in time to understand it better.
Sir, could you please tell us about the meningeal lymphatics and the nasal lymphatics? And if I'm not wrong, it drains to cervical lymph nodes. So any pathology related to these structures?
Not yet.
How does it?
I know about the meningeal lymphatic system that's been described some time ago. And you see, in US research, if a research group gets a lot of political mileage and eyeballs by publishing a large body of work on the glymphatics, the critics are going to look at alternative data which may question some of the facets of the hypothesis that has been raised. So out of those critics, there has been a demonstration of these two key features. One is the meningeal lymphatics. What role does that have to play in clearance of CSF from the brain? What is its physiology? We don't know all the details as yet. We also know that there is this phenomenon of efflux from the CSF, from the vasculature during sleep into the interstitium again. Like I mentioned, that there seems to be a sloshing back and forth phenomenon. We don't know the finer details of that physiology because a lot of it is studied essentially in mice, and then indirect assessments are done in humans. So I'm sure we will learn more about it as time goes by.
Yes, sir. Sir, if you allow, I'll ask my colleague Dr. Dhruv to take further questions.
Yeah, go ahead.
Yeah, sir, is it okay if I take a few questions? 5 more minutes?
Yeah, all right.
Yeah, so I think Dr. Dhananjay Zuberkar wants to know, is there any role of yoga in preventing Alzheimer's disease?
Yeah, so I don't know much about this. I have a colleague, Dr. Shraddha Shah, who works with us in Jaslok, and with Dr. Petrus Wadia and our whole Jaslok team, Dr. Philip Mocha and the others. She does a lot of mindfulness and meditation therapy in patients who have cognitive impairment. I shared with you some of the emerging data which seems to imply that mindfulness, when performed over a prolonged period of time, does seem to produce positive neuroplastic changes in the prefrontal cortices, the anterior cingulate, and also in the morphology of the corpus callosum. We know that these are important structures for cognitive coherence and for optimal cognitive performance and sleep optimization. While this information is relatively new, if more work gets done, we will probably learn more. So about yoga, I'm not sure because I haven't studied it. My interest was in meditation and mindfulness because I think that's emerging as a very big intervention paradigm in the Western world too. And I shared some of those papers. You can also look it up. And if somebody needs those PDFs, I'm happy to send them across.
So in the interest of time, I'll make it a little rapid fire this thing. So Dr. Rahul Kulkarni wants to know, are there any other diseases of the glymphatic system that you're aware of? And a connected question is there any role of vitamin deficiencies in the glymphatic diseases?
I'm not aware of that, so I don't know.
Okay. Sir, Dr. Sudhir Shah wants to know whether the glymphatic system has any role in mood and behavior.
I mean, there is no question. If you listen to the first talk in the series, we know that REM sleep clearly is a great determinant of emotional modulation and mood stability. And there are numerous experiments which have shown this. And we talked about the neocortex and the prefrontal cortex modulating amygdaloid influences on mood and emotion. So while there is no direct correlation with glymphatics, but sleep optimization and the harnessing of an optimal amount of REM sleep clearly has been shown to have beneficial effects on emotion. In fact, the Berkeley group is trying to uh, lobby for starting schools in the US a little later at around 11 o'clock so that children can have a greater amount of REM sleep between 4 and 7 or 8 in the morning, and they can be emotionally more stable.
So Dr. Pramod wants to know any role of the glymphatic system in diseases which affect sleep, such as Iglon 5 or any antibody-mediated disorders.
I don't know really. I mean, this is all new stuff, so this is all fairly new. So
I think I'll end with the most important question: how to prevent insomnia? Somebody has asked this. How do you prevent insomnia?
Well, joking aside, one of my colleagues, Azad Irani, will remember how we discussed this in one of our sleep journal clubs. I would suggest that all of you read Matthew Walker's book, Why We Sleep and Dream. And I would suggest that you go to YouTube and look up Matthew Walker. There's a TED talk by him on sleep optimization. And there are 2 small videos of about 10 minutes each on 5 tips for sleeping better. So if you go to YouTube, look up Matthew Walker and 5 tips to sleep better, it will answer your question.
Okay. I think thank you so much, sir, for your patient hearing. We had a lot of questions. I think Ashish and me ensured that all questions were asked, and we are very happy that you answered them all. I think we can conclude the talk.
Thank you so
Yes,
much.
sir.
Thank you.
Thank you very much,
so
sir. And all would agree that the series is aptly titled Joy of Sleep.
Thank you.
Okay.
Good day.
Thanks. Bye.
Okay. Bye.
Thank you so much.
Thank you.